Okay, good morning. Thank you all for joining us today for what promises to be a terrific webinar. So this is the Collaborative Core Center for Clinical Research Speaker Series, and we appreciate you joining us this morning. We have a uh, wonderful presentation in store, I'm sure, given our speakers today, and we will have two speakers. We will hear both from Liubov Arbeeva, who is a biostatistician in the Thurston Arthritis Research Center at the UNC School of Medicine, as well as Dr. Amanda Nelson, who's a professor of medicine, as well as being adjunct in epidemiology. Also at UNC, she's the director of the UNC CCCR, as well as the director of the Phenotyping and Precision Medicine Research Core within the CCCR. Today, the session is titled An Overview of Clustering Methods for Research, and I'll turn it over to Liubov.
Can you see my screen?
Yes, we can.
Thank you, Todd. And I will cover the first part of our presentation. It will be a little bit statistical, and Amanda will provide you with examples from recent papers and from our research. So I have no disclosures, and I have a few references for you that you can use after this presentation. And the red one is very helpful, and I use this a lot for this presentation. And it is very easy to read, and I highly recommend this for clinicians. A few additional links for you and your data analyst or biostatistician if you wanted to use R packages or Python. So what is clustering? This is machine learning technique, often unsupervised, and clustering aims to group unlabeled data with little or no supervision into underlying subgroups present within the data. Clustering can assist in identifying disease subtypes from clinical data and electronic health records, which will result in more personalized treatment strategies, and more efficient clinical trial design. Gene clustering is widely used in identifying functional groups, so we have a better understanding of disease biology. The purpose of clustering is not only to group unlabeled data, it can be also used to identify outliers, to understand the underlying structure of the data, and to extract meaningful information from unstructured data like images and free text. Originally, all clustering algorithms have been classified as either having a partitioning or hierarchical strategy. It is broader now, and in addition to first two that I just mentioned, there are density-based, model-based, and grid-based algorithms combinations of this, and many extensions. So this is well-developed statistical area. And before I continue, I wanted to refer you to the paper recently published in the Journal of Rheumatology. And if you are interested in common clustering techniques that I just mentioned, you can find a lot of useful information, references, and details, which I cannot cover in 20 minutes today. We will talk a little bit about less common techniques, and I hope it will be helpful. So this is a workflow that I have for myself when I do clustering. And I highlighted 4 steps in red that cannot be performed without clinician or biologist. Specifically, the most important initial step when you ask yourself, is clustering the best choice? Is there any theory supporting heterogeneity in my data? The decision about features that you can use, the clinical interpretation, all these steps require a person with a strong background in the field. And I will talk about the selection of clustering algorithm, the blue part, which is more statistical. So applying a clustering algorithm is much easier sometimes than selecting the best one. So let me cover a few questions that you and your data analyst have to discuss before clustering analysis. So first, what type of data do I have? In ideal situation, we have only continuous variables or only binary like yes or no. In real life, input data is a mixture of different data types and often from different sources like clinical data, images, wearable devices, and so on. In general, k-means works best with continuous variables, and hierarchical clustering can handle a variety of variable types, including categorical variables. But as always, it will depend on your data. Some clustering algorithms work better if all variables are on same scale, like k-means is a good example. Other methods can handle data on different scales. Your data can be high-dimensional, sparse, with outliers and missing observations. Just keep in mind that some clustering methods can address these issues better than others. So let me start with well-known k-means algorithm and explain why it might be not the best option in some situations. The k stands for the number of clusters that we're trying to identify, and we have to specify that number of clusters before we start. The next step is to specify the cluster seeds. Seed is basically starting cluster centroid. It is selected at random or is pre-specified by data analyst based on prior knowledge of your data, which is less common. So then the algorithm iterates through assignment of each data point to the closest mean, followed by recomputation of the cluster means. So now I will talk a little bit about this paper published recently, like a few years ago. And this is a very nice example of where unsupervised k-means clustering was applied to identify osteoarthritis endotypes using targeted metabolomics. So this clustering analysis included only OA patients with 162 metabolites measured in plasma. So the authors identified three endotypes, and one of those clusters showed some association with muscle weakness. And the data suggests that a subset of patients could belong to an inflammatory endotype. This is very interesting paper and emerging topic in OA. But I will point out some statistical details that can be important. So I work a lot with metabolomics data, both targeted and untargeted. So I have to say that metabolites are on different scale. Some vary from 0 to 1. Some can range from 0 to thousands. So, and the data sparse with a lot of zeros. You can have features that are the same for all participants, or features that are zero for everyone except one person. So why did I mention this? Because, I wanted to emphasize that the analyst did a lot of work exploring the data before using k-means algorithm, because, original data is not always ready just to throw this in the k-means. So what I found interesting as a biostatistician is this area where the border between black and orange clusters is really blurry. And it reminds me a few examples from my experience when statistics tell me that there are two clusters, but the epidemiologists say that three clusters have more sense. And specifically when data is small, and so I will go back to this example again and to this picture again. And I also wanted to mention that the data that they used is high dimensional, 162 metabolites. So sometimes it makes sense to reduce dimension, for example, with principal components analysis. So it will reduce and remove the noise and will make the main signal in your data more apparent for k-means. So you can also see a few outliers and some, area like more dense and less dense. So just keep in mind that clustering is data-driven technique. So different technique can drive you to different conclusions even with the same data. So the k-means is very simple, but it comes with limitations. For example, k-means doesn't work well with outliers, like I told you. And when the clusters have different shapes and sizes, the clusters are not nested. So you can do some steps like I described to make data more ready for k-means. And actually, original k-means algorithm has extensions that can improve the performance in some situations, but sometimes k-means might be not the best option. So you can think about density-based options, and it can identify clusters with different shapes and sizes. It is more robust to the outliers. The downside side is that it is very time-consuming. So the next question is, what is the size of my data? And when we think about the size of the data, we almost always remember about power analysis and sample size calculation. In general, power estimation cannot be conducted for clustering analysis. It is exploratory by its nature. But here is the paper recently published with some general recommendations. At the initial planning stage for grant proposal or for the manuscript, you need to ensure that the clustering approach is feasible. A large number of clusters estimated from a small sample of clusters with very small sizes can be indicators of lack of robustness. So here on this slide, I outlined a few recommendations, but you will find more useful information in this paper. So I use it a lot for grant proposals. So please pay attention to the last two bullet points. These are very useful if you have limited sample size and high dimensional data. And I will talk about fuzzy or soft option in a few minutes. So we all know that a small sample size can be an issue, but let's look at this from another angle and think about computational challenges. Hierarchical methods use iterative merging of points based on pairwise distances. So you will need to estimate the distance between each point and each other point in your data. So k-means in general is faster, especially for larger data sets. And this is just because k-means clustering only needs to estimate the distance between each point and the cluster center. So the next question is how we define that two data objects like participants or images are similar and fall within one cluster. So this is very, very important step because most clustering algorithms require measure of similarity or dissimilarity to group observations. If you are using R or other software, make sure that you understand the default option and customize if you think it is more appropriate for your data. So a little bit more math on this slide than I'd like, but I just wanted to impress on you how important this step is. On the left, I have three common measures of continuous variables, four continuous variables, and all of them are just extensions of the Minkowski distance. Look how different the red distances are, and that can affect the results of clustering depending on presence of outliers. If you have high proportion of zeros like WOMAC in the general population. So on the right, I have even more options, and these are only for continuous variables. So just keep in mind that the one can be better for you than the other. So now I will go to the second row, which is more interesting, and this clustering methods not so widely used. So can one object and participant belong to more than one cluster? So most clustering algorithms identify mutually exclusive groups of data, but sometimes overlaps between clusters can explain hidden structure in the observed data better. So now I will briefly mention overlapping clustering methods that tolerate overlaps between clusters. Why? Why? How it can be useful. Let's think about biological applications. We know that genes have more than one function for proteins that participate in multiple pathways, so it makes sense to assign genes to multiple overlapping clusters. In natural language processing, newspaper discuss politics, entertainment, sport, or all. So it can be useful to classify newspapers to multiple news groups. So the next question is, can a data object belong to a cluster with some degree of membership between 0 and 1? Now we will discuss soft or fuzzy clustering. The overlapping clustering that I just mentioned should not be confused with soft clustering methods, where we allow objects to belong to one cluster with some degree of membership. In overlapping clustering or increased clustering, the membership is 0 or 1. Overlapping clustering is less common than more general application fuzzy clustering. But for example, k-means has both options, both extensions, overlapping k-means and fuzzy k-means. So why it can be useful? So we allow for overlapping clusters. It is more robust to outliers. So because of the gradual transition from one cluster to another. Soft clustering provides more detailed understanding of the data compared to crisp and overlapping options. And what is very important, it can provide higher power in multivariate normal distributions. So in hard clustering, a disease can be present or absent, red or blue. In soft clustering, the disease could be of some grade red and some other grade blue. And you can think about many examples from medical field. For example, hyperplasia can be defined as normal endometrium or abnormal endometrium by two different physicians. Another good example is KL grade. So the same image can be classified as KL grade of 2 or 1 by two different radiologists. So this is a very simplified illustration of differences between hard and soft clustering, but in real life, the estimation of this degree is difficult and very mathematically extensive algorithm. So let's think about why fuzzy clustering. And now I wanted to go back to the example that we discussed a few minutes ago. Clustering and endotypes. Remember that there are more than 600 OA participants, so in some situations it is a luxury. For example, if the biomarkers are expensive or if you study very rare disease. So the biomarker clustering performed by authors can be used to stratify patients with OA into groups with distinct endotypes. So in the future, this approach could potentially drive clinical trial certification and serve as the basis for precision medicine strategies for OA. But let's look at this border again. Would it be possible that one person can be at high risk for both CHD and osteoporosis? Is there anything special about his profile? What if I have smaller data, a couple of hundreds only, and wanted to replicate these results in my sample. So there are like many situations when fuzzy clustering is more robust and than traditional k-means, specifically for heterogeneous diseases like OA. So this is another very nice illustration to show that soft clustering is more flexible and robust than a hard one, because biomedical article may cover several medical disciplines and issues. So imagine that we have n medical papers and v k-words resulting in n by v matrix in the upper right. So each row is a paper and each cell is a count. So we assume that there are k topics, for example, radiology, pediatric, autoimmune, obesity. The key idea is that each paper can be represent multiple topics. So we wanted to discover a topic distribution over each paper and word distribution over each topic, which can be represented by two matrices on the left, n by k and k by v. So, and Amanda will show you the example from our research when we applied this epidemiology to microbiome data. And our rows were like participants' microbiome profiles, and the columns were taxa. So next, is it possible to find clusters in a subset of features? Yes, and it can be done with simultaneous clustering of the rows and columns of your matrix. I'm moving to the last part, by clustering. The main idea is that different subset of features or columns can be informative for some rows, but not all. By clustering algorithms were originally developed for gene expression data. So let's say you have a microarray represented as 15 by 10 table. So we have 10 conditions and 15 genes. So we ran biclustering algorithm, which revealed 5 biclusters. Each bicluster on the right is a subset of genes and a subset of conditions. So now let's think about that from another perspective. Your rows are patients, and the columns are biomarkers or tests. They can be expensive, invasive, painful. So you wanted to identify disease phenotypes using biclustering, and now you can see that a subset of biomarkers can be informative for one disease phenotype, but not all. So there's no need to collect all expensive tests from all patients. As for all clustering algorithms, you can use all types of data, omics, phenotypes, even results from previous analysis. For example, betas or odds ratios from different GWAS meta-analysis or fold changes from gene expression data and so on. So look at this red circle. You can use effector genes for several individual diseases like type 2 diabetes, CVD, obesity, OA, from GWAS summary statistics available in publications for free, and apply biclustering, soft clustering, or even simple methods. So, as you probably understand now, there is no consensus on how to select the optimal distance measures, the clustering algorithm, and the number of clusters. So in real life, we apply different methods, select different parameters, and of course, validate clustering results using external and internal criteria. So external criteria, this is very important. I just want to mention this. Compare the results with the known cluster structure. So they can be used with a theoretical framework to compare different methods. And they can be also used to estimate the stability of cluster assignments over resampled data when you do internal validation. So there are like few examples on this slide in the third bullet point, and the reference when you can find a lot of information about external criteria. So internal criteria are less common and they are similar to goodness of fit indicators. And many of you are familiar with relative criteria that can be used to select the best number of clusters. And examples are Dunn index, elbow methods, and many, many others. As always, results from by-clustering should be reported, for all clustering, should be reported with sufficient details, including nature of data, theory supporting possible subgroups, detailed analysis procedures, and so on. So here's the checklist from a key reference paper that was the red paper that you can use for your manuscript, and it is very useful. So not only for people who publish paper, but for the reviewers as well. So the last slide is with my acknowledgement. So I wanted to thank you, Thurston Arthritis Research Center and the CCCR core and all my funding agencies that support my research. Thank you.
All right, how's that look?
Looks fine.
Awesome. So first point, get yourself a really smart biostatistician if you want to do this stuff. So I'm going to give you some examples of different ways that you might apply some of the methods that Liubov just went through. None of these are really relevant. So we're going to talk about soft clustering in EHR data. We're going to talk about bi-clustering in clinical data instead of just genetic and sort of basic data, and then the compositional microbiome data that Liubov mentioned. So first, this is a nice example that's just kind of general, I think, understandable for kind of people at all levels who are trying to get their head around what fuzzy clustering is. And so this is an EHR-based example. And what this group did was they collected data from their EHR from 2017 in a single hospital. It had 22,000 patients. And the criteria were really that they were getting outpatient care and they had at least 2 chronic diagnoses. You'll see why in a minute. And they wanted to identify clusters of medical specialties. So it was kind of a health procedural kind of study. They wanted to know how many specialties were being involved in their hospital care, but it's a really nice illustrative example. So they used fuzzy k-means. So as Liubov mentioned, this allows clusters to have groupings of people. So you don't have to just be in one cluster. You can sort of have partial membership in multiple clusters. A patient could thus belong to several different clusters, seeing different medical specialties, and have a sort of proportional membership in those. And that's kind of what the group wanted to know. Additionally, the medical specialty could be part of more than one cluster. So it really allows all combinations across both sort of levels of clustering, which in this setting and many other clinical settings is a little bit more meaningful because we kind of think of things on more of a spectrum or a grayscale rather than just black and white hard clustering style. So for the specialties, they found 6 clusters. I just illustrated 2 of them here. So they had a big cluster of people that were just coming for dermatology, plastic surgery, kind of turned out to be skin cancer sorts of folks. Cluster 2 was sort of more complex, seeing 6 specialties. This one included rheumatology, so I underline that. But you can see in the bottom figure, there's sort of an even spread across multiple different specialties. So they're getting a lot of different care. Then cluster 3 was really lung-based. Cluster 4 was kind of cardiology and geriatrics. 5 was neurology, anesthesiology, kind of a pain group. And cluster 6 was kind of general internal medicine. So these makes sense. Again, it's a certain hospital environment and a certain group, but this is the illustration of the fuzzy clusters, which now you can put back into the theoretical model that Liubov presented. And so you might have a cluster like this, where almost everybody in the cluster is really only in that cluster, and that cluster is sort of self-identified, right? So it's basically only the skin cancer patients, as it turns out. You can read the paper and see all the details. But there's also clusters like this where people are really evenly distributed across multiple clusters. Maybe they're seeking care for multiple conditions, seeing different groupings of specialists at that visit or over multiple visits, and then everything in between. And it really allows that variation, and it is a nice visualization of how to look at that. Okay. And we're going to go to biclustering. So this is the one Liubov described in the genetic data. So kind of looking at clusters of subsets of data. And this example uses data from the baseline OAI visit. So 6,000 knees from 3,000 people and 86 features. It uses this specific biclustering algorithm. There are several, and they have different strengths and assumptions, as all models do. So kind of understanding which algorithm you want to use. And we identified 6 mutually exclusive biclusters. I'll explain what those look like in a minute. And then we're able to look at those biclusters or phenotypes or whatever subgroups you want to call them with outcomes. So the biclustering was done just at baseline, but we looked from baseline based on the biclustering at what happened to those people later on. We were also able to validate the clusters. This is one of the things Liubov was talking about, sort of how do you determine if the clusters are real, if they would be there, in other samples, using this SIGCLUST procedure. And so again, just to revisit why biclustering versus regular clustering, in this example at the bottom, if you just have this heat map, right, kind of random measurements and knees and who knows what's going on there, it actually looks just like that if you do it in OAI. Biclustering, in contrast to clustering, which is in the middle, allows these sort of sub-matrices, right? So you can look within a certain group of knees at a certain group of variables and come up with a little sub-matrix within the data, rather than assuming that all measurements are the same across all knees within a given cluster. So it's just a little bit more subtle, and it might be a little more clinically relevant. And so this is the OAI data. Is just a big visualization. Again, the reference is there. And there were 6 biclusters and a very few knees at the far right that did not fall into a bicluster. And I'm going to describe primarily 4 and 5 here. You'll see that there's gray marks. The gray marks are variables that were not important in that bicluster, right? So again, it's these submatrices within the data within the knees. And if we look at those 2, 4 and 5 here, we see that they are characterized at baseline. So this is the data that developed the bicluster by poorer physical health, more pain and symptoms by KOOS and WOMAC. The WOMAC is flipped, so they're all on the same scale. And also for continuous variables, but more likely to have knee injury, more likely to have other joint pain, more likely to be Black versus white in this cohort. So kind of indicators we know might be relevant for poorer outcomes. Again, this is just at baseline. So then if we take those biclusters developed at baseline and look at the outcomes, which were not used in the algorithm, which is really important, right? If you sort of throw in all the variables, including the outcomes, then you've kind of biased your results. So these features were not included in the biclustering algorithm itself. But if we now look at those 2 biclusters that seem to have the poorer prognostic features, we see that they're also more likely to have OA, they're more likely to have progressive OA, they're more likely to end up getting a knee replacement at 48 months. And then 96 months, here's joint space width loss, so narrowing of the joint space, more common in the 2 biclusters, and also more likely to have poor WOMAC trajectories. So kind of all fits and makes sense. And the idea is not that this is some dramatic groundbreaking thing, right? We could have kind of guessed this, but it's sort of all in one analysis. And it might give insights in a different data set into new phenotypes that perhaps we hadn't thought of. These are just some of the other plots you can get out of this. So if we just look at bicluster 4, we can look at each individual variable and the distribution of that variable within the bicluster versus that variable compared to the overall population. So you might get a sense of certain features that are driving that association. And then this is the SIGCLUST diagnostic figure that sort of shows that bicluster 4 is in fact distinct from the other biclusters, as is 5. some of the others are more overlapping. So it's kind of a good sanity check. Like, yes, indeed, there are more than one group within our data. Okay, so now we're going to move to our microbiome composition data. So I've got two examples here, and you'll see kind of how this relates back to Liubov's talk in just a minute. So we're going to talk about Dirichlet multinomial mixture clustering and its application to microbiome data. So this is 145 RA patients. So now we're in RA, reset our minds a little bit. And they have demographic and clinical features, fecal samples characterized by 16S. This is kind of the older technology, but it's also much cheaper. And they want to look at relationships between the groupings of microbiome data and their clinical measures, and whether those then associate with clinical response to treatment. And so this is very similar to the figure that Liubov showed with the papers and the subject areas, but now we're looking at participants and genus information for the microbiome, as she mentioned. But we're doing the same workflow with these compositional data. So we assume that these are present in all participants at varying proportions, and we can kind of cluster those based on that. And we're going to call those enterotypes in the end. These are, again, at genus level. We're going to divide the RA patients into different community types based on sort of their levels of these different genus. And this is fine. There are a kind of like, anything else like PCA with a scree plot, there are ways to identify how many clusters, how many components, how many enterotypes probably are in your data. There are more than one, so you often will do multiple of these to figure out what the best fit, what makes the most sense, what's the most parsimonious, etc. So they use this Laplace value and said, okay, we have two distinct Dirichlet components, and they're going to call these E1 and E2. And this is kind of the plot. It kind of looks like a k-means, it's not, but it reminds me of that. So we have this blue cluster with its centroid up here and this red one that's down here. And now we're going to look at different characteristics of those two clusters. So here's E1 and E2. E1 is red, E2 is blue. So if we look at E1, we see this larger yellow area. Those turn out to be Prevotella. And if we look at E2, we've got more Bacteroides. So now they're going to call E1 kind of the Prevotella dominant and E2 more Bacteroides dominant. And they developed an enterotype-based gut microbiome human index, which they call EGMI, thank goodness, so we don't have to keep saying that, which is basically a score based on this figure. And we're able to apply that to independent RA data sets. So this is, again, this important aspect of validation, both within your data, but also externally. And they want to sort of show that that EGMI score could classify patients according to this kind of enterotype. And they found here on the right that the E1 group here in red was more likely to respond to methotrexate, while the E2 group had a higher proportion of methotrexate non-responders, right? So that's important. If we could identify those ahead of time, we might know which therapy would be beneficial. Again, there are a lot of other things you can do. So if you look at supplemental data in these kinds of papers, they characterized immune cells by the two enterotypes. They looked at inflammatory markers. There's all the different bacterial genus that were involved in the two enterotypes. So maybe you're not interested in Bacteroides, but you're really interested in Ruminococcus, which has been described in a variety of different autoimmune conditions, and you want to look at that. So, if your data doesn't include the primary result here, it may still be in the additional information, which is why it's so important to report all of the results, even if it's in a supplement, so that people can replicate and reproduce your data. And these are just some more diagnostics for the EGMI score. And this is our recent work that used that nice protocol that I showed you earlier. Cameron was a medical student with us. He's now an orthopedic resident. And here we're looking at OA. It's a small sample, it's 90 some, I think. But we can look at the distribution of the enterotypes again. So what are the enterotypes? There's 5 of them. We use those diagnostic plots you just saw. And you can see the larger dots are have a higher representative of that genre. So 4 has a lot of Bacteroides. 3 has a lot of akkermansia, et cetera. And then we can look at the proportion within the population that have those enterotypes. So 4 is kind of the common one, and then 5 is sort of a little bit less, kind of 50% here. And then we can take the enterotypes as clusters now, right? Instead of all these individual bacteria and multiple testing, et cetera, and just do these 5 tests to say, well, are these enterotypes associated with important clinical characteristics? And we see that 3 and 4 are actually more likely in controls. We see some race and gender differences and some differences here by biomarker levels. So those might be hypothesis generating for future work. Okay. So clustering can be useful to understand similarities and differences among subgroups. And it's really subgroups of observations, subgroups of data types, again, this idea of, can we not do particularly invasive or expensive testing because we know that we can identify those subgroups using other modalities. There are a variety of different approaches. They all have different assumptions. So you do need to work with your biostatistician from the beginning to really understand what your data are and what approach is going to be the most suitable for those. These novel clustering methods can improve our understanding of complex subgroups, which all clinical data is complex, right? So thinking beyond just k-means to, what would really help me understand what I'm dealing with here? And there are many more than we were able to talk about. And again, I'll just keep saying to work with your biostatistician, because there's just so much going on with these, and it's easy to go wrong if you just sort of plug it in and hope for the best, right? These are the references that I showed. And these are our acknowledgements. So I will stop sharing so that we can go to questions.
Okay, great. Thank you both so much for that terrific presentation. We do have time for questions, and so I welcome anybody who has a question, preferably to unmute and ask that question, but we'll also take a look at the chat if you want to put anything in there. And I'd like to kick off the questions with maybe a bit more technical question for Liubov, and that was, you referred to a paper dealing with power and sample size for some of these techniques. And I'm not asking so much about specifics for what was in that paper as opposed to just the general concept that applies in the sense that there's not really maybe a p-value or formal testing
Yeah.
or a confidence interval width or something like that. So what is the notion of power? How does that play into these types of methods?
So I wouldn't call this power. It is more estimation of a sample that is feasible for your approach. So like they recommend like at least 30 people in a cluster that are like when you start the analysis in the proposal or in the manuscript, you can say that, my expectation is that I will have 4 groups. So you will need more than 100 for sure. And, otherwise it's like, and if the sample is really small, so they have some recommendations how to deal with that. So dimension reduction, fuzzy, soft approaches, and everything else. They have a lot of simulations so in this paper. So this is a very useful paper. So, and me and Amanda, we use a lot of these methods in proposals. So I'm always looking for new publications in this area, and they kind of appear more and more. They're like, so like 5 years ago, there was nothing. So you cannot say anything to justify and to, make sure that reviewer will say, okay, your sample is enough. And, it is very important because, we always, sometimes you have clinical electronic health records but sometimes it's so expensive. You cannot just like afford more. But you can say that, okay, it's small sample, but I know how to deal with that and I will do this. And reference the paper, yeah.
Great.
So for the last paper with CAM, there was also a recent publication how you deal with this with Dirichlet regression. Yeah, so it's like for each method you can, so with simulation studies for power and sample size estimation, so you can, look for new publications. People work on that.
Great. Thank you. I'm not seeing anything in the chat. Again, feel free to unmute yourselves or if you want to raise your hand. And again, while we're waiting for other questions, again, maybe a bit more on the technical side, but can you speak a little bit to software considerations? Like, are people mostly implementing this in, say, R packages, or what kind of software are people using for these kinds of
So
methods?
R is very flexible. So I use all R in like 90%.
Hmm.
So by clustering, Jenkins Church was done in Python because it was Tom, our wonderful student. You will never believe, and that was his main language. But I know that Jenkins Church is available in R as well. So if I will use it, I will use R. I like it more. But K means of course, and fuzzy, all because it's a lot, and you have to find the one that fits your research question.
Thank you. Brooke, I see that you placed something in the chat. Did you want to ask that question?
Or can you hear me?
Yes.
Okay. You mentioned the RAND index as a way to kind of compare clusters once your analysis is done. What's the benefit of using the RAND index or the adjusted RAND index? Is there a situation where one is more appropriate than the other?
I usually use several indices, so I never use one. And for most papers that I was included as a co-author, I use like at least five to make sure that they all in agreement. And if you email me and ask me, I can send you the R package that creates at least 7 indices and plots in the one plot. So because some indices they have to increase and some to decrease. Yeah, so it's not always in agreement, but I usually choose the solution where more indices are in agreement. So it's like, yeah, it's not always, as easy as in, statistical books.
Thank you.
Yeah.
But I use
I
adjusted more than rent index.
would chip in that that's a place where you can have some clinical input too. So often we'll look at all of those different diagnostic
Mm-hmm,
plots and be like,
mm-hmm.
well, this one says 2, but we could do 3, and then kind of, what makes the most sense, which again, most parsimonious. So, it's really iterative and thinking about all
Mm-hmm.
these different aspects rather than just doing one because it's one that you've used
Yeah,
before or
it's
something.
the same as with latent class analysis, latent trajectory analysis. We never look at BIC only. We use several indices to make sure that they, tell us the same. Sometimes it's not, but.
Great. Other questions? Well, if not, I'd like to take the opportunity to thank both of our speakers, Liubov and Amanda, for a very informative and educational presentation. I'd like to thank everyone who attended, and I'll remind you that we do record and archive these presentations, that if you wanted to refer to it or share this with any of your colleagues, we use the UNC CCCR website for that purpose. So thank you everyone for joining us, and hope you have a wonderful day. Thank you.